Nutrition and brain development. The first years of life are a period of remarkable brain growth and development. A baby's brain triples in size by its first birthday, reaching 90% of its adult weight by the age of 8. The young brain must produce large quantities of myelin, the material that insulates neurons and allows them to communicate quickly and efficiently. Neurons communicate at connection points called synapses, which are essential for perception, learning, and memory. During peak growth periods, the young brain produces up to 2 million synapses per second. A child's diet must supply energy and materials to support this incredible growth. Key nutrients provide the building blocks for lifelong intellectual, emotional, and social development. Breast milk is the gold standard for infant nutrition, and it is impossible to replicate its exact composition. However, nutrients found in breast milk, known to promote brain and eye development, can be added to infant formula to support the baby's developing intellect. These nutrients include phospholipids, fatty acids, taurine, choline, zinc, lutein, and iron. Phospholipids and fatty acids. Phospholipids are a type of fat found in the membranes of all cells and in myelin. The unique polar structure of phospholipids makes them essential for membrane fluidity and cell signaling. Fats make up 60% of the brain and nerves and 75% of myelin. During the first year of life, the fat content in an infant's brain more than triples. 2/3 of this fat consists of phospholipids. Together with essential fatty acids, phospholipids are the primary constituents of cell membranes. Two particular fatty acids, docosahexaenoic acid, DHA, and arachidonic acid, AA, play a strong role in cognitive and visual development. DHA and AA are highly concentrated in neural membranes, where their flexible structures enhance the cell's signaling functions. Outside the cell membrane, DHA and AA promote the growth of axons and dendrites and the formation of synapses. DHA is also prevalent in the outer segments of photoreceptors in the retina. DHA's flexibility facilitates the movement of pigments in these light-detecting cells. Taurine and zinc. Taurine is a physiologically indispensable amino acid present in high concentrations in the developing brain and retina. Taurine in infants is considered a conditionally essential nutrient. In the eyes, taurine contributes to retinal development, particularly in the cell membranes and outer segment of cone receptors. It may also be needed for normal auditory response. The mineral zinc is a critical component of several enzymes involved in myelination and neurotransmitter production. Zinc helps regulate the release and uptake of neurotransmitters as well. Taurine and zinc both help to regulate membrane permeability, protecting brain cells against excessive swelling and shrinking from osmotic changes. Eye health. Taurine, DHA, zinc, and the antioxidant lutein are important for eye health and are all highly concentrated in the retina. DHA gives retinal cell membranes the fluidity they need to function well, but also makes them vulnerable to damage from light and free radicals. Lutein is the main carotenoid found in the retina. It is highly concentrated in the macula, a distinct yellow spot in the central portion of the retina. Scientists believe that the macular pigment acts as an antioxidant to protect retinal membranes against light-induced damage. Choline. Choline is an important nutrient that is required to produce the neurotransmitter acetylcholine, an important messenger in the brain. Choline is also needed to synthesize certain phospholipids. Iron. Iron is a mineral that is necessary for basic neuronal processes, such as myelination and neurotransmitter production. It is also necessary for transporting oxygen in red blood cells. In summary, a variety of nutrients work individually and in concert to support the developing brain and senses. When added to infant formula, these compounds can help promote optimal brain growth and lifelong mental development.